Hi guys, I am Sarang Anantrapawar. Today in this video we are looking the information about the enzymes. Then first what are the enzymes? Enzymes are the biological catalyst which speed up the biochemical reaction. Enzymes may be the global three-dimensional globular protein, and some of the RNA species also act as the enzyme, that is called as the ribozymes. These are the enzymes, basics of the enzymes. It is the catalyst which acts as the catalyst which speed up the biochemical reaction, and in some time it should be the three-dimensional globular protein, and the RNA, some special RNA species also acts as the enzyme, are called as the ribozymes. The structure of then what is the structure of the enzymes? Enzyme structure is composed of the active sites, that is active sites, cofactor. These are the enzymes. The structure of the enzyme made up of the active site and cofactor. The active site of the enzyme is the region that binds to the substrate, cofactor, and prosthetic groups. The active site occupy the 5% of the total surface of the enzyme. An active site has the specific shape, uh, which change in the shape of the proteins, change in the active site and the function of the enzyme. Then there are two types of active site, that is the binding site and the catalytic site. The binding site is the choose the substrate and bind to the active site. Binding site choose the substrate and uh, bind the substrate and at the active site, binding site. And after that, the catalytic site is it performs the catalytic functions of the enzyme, which function in the that is the speed up the biochemical reaction. Then second is the cofactor. It is the non-protein part which carried out biochemical reaction and converts inactive protein into active protein. Then what is the there are two types of the cofactor. First is the organic cofactor, that is the organic, that should be the organic molecule. uh, Example of the organic cofactor is the glycogen phosphorylase. And second is the inorganic cofactor, the that are the inorganic molecule. The example of the inorganic cofactor is the hexokinase. After that, the what is the substrate is the reactant in the biochemical reaction. It is known as the substrate. If the substrate is if the substrate joins to the enzymes, when it gets the into the substrate enzyme complex. Substrate is joined to the enzyme site, binding site, then it forms the substrate enzyme complex. The site of enzyme synthesis, the enzyme is synthesized by the ribosomes, which are attached to the endoplasmic reticulum. Information for the enzyme is synthesis by carried out by the DNA, and the amino acids bounds together to form the specific enzyme according to the DNA code. And there are two types of enzymes: intracellular enzymes, that are made inside the body and works for the inside the that are sorry the intracellular enzymes are the enzymes that are made uh, intra, inside the cell and work for the inside the cell functions. That are the example of the intracellular is intracellular enzymes, that is the oxido reductase, which helps in the oxidation. And second is the extracellular is the enzyme synthesized by the cells for the external working. The example of the extracellular enzymes are the digestive enzymes secreted by the pancreas. After that, what are the characteristics of the enzymes? In uh, enzymes are the enzymes speed up the biochemical reaction. These are the does not affect the end product of the biochemical reaction, and they are highly specific in their to their actions, and they are sensitive to the temperature, pH, uh, and substrate concentration. This is the main characteristics of the enzymes. Thank you so much. We will meet soon.